So again, remember there are 3 DNA binding motifs we just talked about. Zinc finger motif is one of them, and then helix turn helix, okay, and then coiled coil motif. 3 motifs, or 3 domains, DNA binding domains we just talked about, okay. And then a lot of proteins function as enzymes instead of cells. So let's talk about enzymes. Well, how they maintain their structure and function. Okay. So enzymes, as you know, are catalysts. Catalyst in their definition, they are not used up during the process, chemical process. Instead, they are speeding up the conversion of chemicals. That's what they do, right? So how do they speed up the chemical reaction? In physics, by doing this work, by lowering the activation energy barrier. Okay, so that's the activation energy from higher energy state to lower energy state. This conversion can take place, but to prevent that happening spontaneously, there is a little bit of energy requirement to run this reaction. We call that activation energy. But if your substrate binds to the enzyme, that activation energy is lowered, so this conversion can take place more readily. That's what enzymes do. So how do they do that? How do they lower that activation energy? Normally, enzymes have a direct binding with their substrate. Normally, enzymes are large proteins. Okay, if this diagram is an enzyme, this purple ball, they have active site where they can bind with the substrate. So if this is a substrate, between the enzyme and the substrate, there's a structural fitness, right? And when they bind, come in close contact, these things, H and plus, minus, NH, all those things, they form those non-covalent bonds, like plus to minus. There's an ionic interaction happening. H's may be the hydrophilic hydrogen bonds among the hydrophilic side chains, and there may be other interactions, like maybe hydrophobic interactions as well. So hydrogen bonds, ionic interaction, or hydrophobic interactions, all those things happen between the substrate and the enzyme. So through those interactions, the structure of the substrate is changed, and that structural change is mediated by your enzyme. So once that change of the substrate is made, then substrate can go through the chemical reaction more readily. So that's what's happening to lower that activation energy. Okay. So the binding of the substrate to enzyme causes the change in the distribution of electrons, the chemical bonds of the substrate, and ultimate cause the reaction that leads to formation of product. Right? That's what I just said, right? So active site has a unique geometric shape that is complementary to the geometric shape of a substrate molecule. Of course, that's the fitness we're talking about. Similar to fit of puzzle pieces, yes. And this means the enzyme specifically reacts with only one or very few molecules that have very similar structure. That's the specificity we talk about when you describe the enzyme and substrate interaction. So it's like a lock and key. They have the fitting structure unless they will not open up, right? So that's the structural fitness we're talking about in the active site of the enzyme. And induced fit kind of explains well what changes are made on the substrate once they bind with the enzyme. So substrate plays a role in determining the final shape of an enzyme, and that enzyme is partially flexible. Okay, and when they interact and that the changes are being made, it explains why certain compound can bind to enzyme but do not react because the enzyme has been distorted too much, and other molecules made too small to induce the proper alignment and therefore cannot react. Only the appropriate proper substrate is capable of inducing the proper alignment of the active site. So that's the specificity we talk about. Okay, so yeah, enzymes have unique structure, and then that unique structure allows the binding of unique substrate molecule. Okay, and the regulation of protein activity through binding with ligand. Ligand again is a molecule that binds with protein, and then ligand and protein have very structural fitness, just like enzyme and substrate. Okay. So ligands are molecules that bind a protein in a specific way, just like enzyme and substrate. And ligands are normally small molecules. Ligands can regulate the activity of a protein by stabilizing a particular state of protein structure. Example given here is a lag repressor protein found in bacterial cells, lactose repressor. Okay, this is a DNA binding domain. We'll talk about it again when we talk about the gene expression regulation mechanism in prokaryotic cells. But this protein repressor binds on the promoter sequence, operator sequence of the bacterial genome. Normally, when it binds, it stops the transcription because, as the name says, represses the transcription of a gene because by blocking the access of RNA polymerase. But this protein, when it binds with the ligand, in this case a lactose molecule, coming here and binding on this repressor protein, that shifts the structure of the entire repressor protein, and the structural shift makes a significant change of the entire protein structure, and that change will make this protein fall off from DNA, so that it doesn't bind with DNA anymore. Okay, so we call that allosteric change. Okay, so allosteric change is what? Change happens one side of protein, makes a shift of the entire conformation. So some drastic changes happening on another side. That's what allosteric change. So the sugar binding here change the structure significantly, so the DNA binding happening here is falling off. Okay, that's what it's described. Okay, if once it's binding with the sugar and it comes off from the DNA. So you see, this is the DNA binding domain, and this may be a kind of activation domain binding with the sugar. So DNA binding, you see the helix turn helix motifs there. That's how it recognizes and binds on the DNA. Okay, so the regulation of the protein activity is possible through ligand binding. So what's been regulated? So DNA binding ability without the ligand. Okay, and then that ability is controlled by ligand. When it binds here, the structure change disallows its binding with the DNA anymore, so it falls off. So there's activity regulation just took place. Okay, so that's the end of this lecture. We reviewed a lot about the protein structure, amino acid, what chemical interactions are controlling the protein structure and activities and all those things. All right, thank you.